Welcome. Hello, everybody. Welcome to the History of Diving Museum. We see members, we see some new people, and we have a wonderful audience on Zoom this evening. So thank you for attending and coming to our third Wednesday Immerse Yourself program at the museum. We also would like to thank our sponsors this month. We have our board member, Patty Gross, and her wonderful husband, David, as our sponsor. So thank you very much. The History of Diving Museum has been busy. Last month, we were at the Art Walk. And at the Art Walk, we actually had the Lionfish Central Lionfish Patrol submarine project going on. So everybody got to find out about what they're doing for lionfish research and testing it out and being able to help clean up our reefs and fill up our restaurants so we can be eating lionfish on the menus, right? That's a good thing, and it's a lot easier than going out and catching them yourself. So I appreciate those efforts. On Sunday, the 25th, for those of you that are in town, or if you want to also sign up for Zoom, we've got a author signing and discussion with Dr. David Schiffman. He's going to be talking about his recent book, Why Sharks Matter. So whether you're a diver or not, it's interesting. It gets a lot of people involved to find out about all the various research and different projects that are going on to build that awareness. Next month's Immerse Yourself program is we're going to flip to a military theme. So it's going to be the frogmen of World War II. It's very interesting. And these are the guys that eventually became the Navy SEALs. But this was when all they had was shorts, swim fins, a mask, and a lot of gumption to do what they needed to do. So we're going to find out about that through a military journalist and author, Andrew Dublin. So be sure to sign up for and watch our newsletters for next month's Immerse Yourself. So that's what's been happening at the History of Diving Museum. We say don't drive by, dive in next time you're in the Florida Keys. We've got a wonderful Sponge Hunters featured exhibit that's only going to be open through December 31st. And part of that, in the research and things that we put together for the exhibit, we want to thank Dr. Shirley Pomponi because she's amazing. Not only is she in the Women Diving Hall of Fame, she's an ocean explorer. She's a phenomenal individual. She's friends with more than one of our board members, because we got a couple in the Women Diving Hall of Fame. Dr. Sally couldn't be here tonight, but she's on our Zoom presentation. But Dr. Pomponi is going to talk to us about the medical research and the things that are going on outside of the sponging industry, just collecting them and harvesting them, but actually medical properties and some great new research. So Dr. Pomponi, thank you very much for being at the History of Diving Museum and letting us.
Thank you. Okay, there we go. Well, thanks very much, Lisa. Can you see me okay?
I can.
You can see me okay? Great, thanks. And I also want to thank Patty and David Gross for sponsoring this lecture tonight. And I also wanted to dedicate it to John Stevely, because John Stevely was a Florida Sea Grant extension agent and really an expert on commercial sponges. And all those sponges that I loaned you for the exhibit actually were John's, and he gave them to me, and he recently passed away. So this is dedicated to John. I learned probably everything that I know about commercial sponges from John. So anyway, I'm going to talk to you tonight. We're going to do a little refresher on sponges. What are sponges? And because it's probably been a while since you had biology in high school and college. And so I want to do a little refresher on that and why sponges are important. And then talk a little bit about medical uses of sponges, and then finish up with some of the work that I'm doing in terms of production of these molecules using cell culture. Okay, let's see if that's it here. Okay, so what's a sponge? A sponge is an animal. It lives on the bottom. It's attached to the bottom for its adult life. And so there are lots of different kinds of sponges. They showed up in the fossil record more than 600 million years ago, so ancient animals. There are more than 10,000 species of described species, but we estimate that there are probably another 25,000 that are yet to be described. In fact, even when we dive here off of the Florida Keys or in the Gulf of Mexico, we find species that are new to science. They live in all depths of the ocean, from shallow intertidal down to the deep sea, from polar to tropical seas. And they're dominant components of many benthic marine ecosystems, especially in the tropical Western Atlantic. Sponges are very diverse in the tropical Western Atlantic. There are even some freshwater sponges. They live in freshwater lakes and streams. And some of them can survive out of water for short periods of time. So up north, where, and in fact, when it gets very cold and dry, some of them have these little, almost like spores, and they can survive out of water for short periods of time. In fact, just recently when I was snorkeling in the Indian River Lagoon, I saw a sponge that was pretty high up on a mangrove root, and it was low tide, but this sponge was definitely exposed. And sponge cells can be frozen for many years and they will remain viable. And so I want you to remember that, because later on I'm going to tell you about how we take advantage of that property. So sponges, I mentioned, are ancient animals. They don't have any tissues. They don't have any organs. They're at what we call the cellular level of organization. So they have cells that differentiate to do different functions. So some of them make these little microscopic skeletal elements that are called spicules. Some of them make fibers. And the picture on the right, the diagram on the right, is kind of like a little chunk out of a sponge. And you can see kind of just the general plan, the body plan of sponges. So they've got kind of an external, it's called like an epithelium, but a panacoderm. And then there's a series of these little chambers called choanocyte chambers that are very special, because that's what sets up the water current that flows through sponges. So what makes a sponge unique from other animals are these little collar cells. So they have like a little whip, or it's called a flagellum, and they are in a little chamber, like a little sphere, and they line that chamber and they set up a water current, and that causes a unidirectional water flow through the sponge body. So sponges occur really all over the world. They're very widely distributed. And just recently, probably in the last maybe 20 or 30 years, maybe even less than that, it's been discovered that there are areas in the deep sea where there are these sponge aggregations or sponge gardens, sponge grounds, where lots of sponges occur. And this is just an image of the Atlantic, and every one of those white dots is an area where a sponge aggregation has been found. And this is what it looks like in the deep sea. So they can be, all those little white balls are sponges. And some of them we suspect that some of them may live for hundreds of years. And in fact, they're threatened because they're kind of fishery practices where they're trawling along the bottom, and all of these sponges get caught up. It's bycatch. And it might take decades or even centuries for these areas to recover. And then also with deep sea mining becoming now more important, a lot of the mining, especially like the drilling muds, affect these sponges as well. But sponges can pump a lot of water, like large volumes of water through their bodies. And some of the larger sponges can pump a lot. And there are some sponges that can pump up to 50,000 times their volume. It's kind of the equivalent of a human being drinking about 1 million gallons of water in a day. So it's a lot. And I'm just think about that, because think about, like, for those of you who may have been snorkeling in Florida Bay and you see those big loggerhead sponges, or if you're out on the reef and you see the big barrel sponges, those are filtering a lot of water. And think about how that's kind of clearing out and cleaning up and the water. It can also be a problem because when you have harmful algal blooms, they can get those pores can get clogged, but some of them are really good at kind of filtering out those harmful algal blooms as well. So what we say is that sponges provide really important ecosystem services, like filtering large volumes of water. They're very diverse. That biodiversity is really important. I'll talk about that in a minute. They're more recently, it's been discovered that they're really good cyclers of carbon and silica and nitrogen. So really contributing to that global carbon budget, silica budget, nitrogen budget. So really key for nutrient cycling. And we didn't know that until just a few years ago. So again, that's what makes these deep sea sponge grounds and even shallow water reef areas so important in terms of nutrient cycling and the global carbon budget as well. But what I've been working on for many years is what's called blue biotech. So what are some of the other properties of sponges that can be useful for humans, for human applications? And so this diagram was made a few years ago. It's 7,000 plus sponge-derived chemicals. I think it's probably closer to 9,000 now. But they also have a type of collagen. So some sponges are aquacultured, and the collagen is extracted from it. And the biosilica is also really important because it's discovered that you can make these little matrices that can be used for bone and dental implants out of this biosilica. And in terms of biomimetic inspiration, some sponges make very intricate skeletons, and they can withstand really strong currents. And so that structure, the silica, the spicular structure, their little skeletal structure, has been an inspiration for architects actually to build buildings that can withstand like strong winds. Why is that not working? Let's see if we can advance that. I don't know why it's not working. Hold on a sec. Let me see if okay, there we go. Okay, so you already know, or maybe you don't, but you already know, I'm sure everybody's taken an aspirin. There are many medicines that come from natural products. Many of them are from plants, so plants that live, on land, terrestrial plants, or microbes. So aspirin comes from the willow tree. Morphine comes from poppies. Taxol, a cancer drug, comes from the Pacific yew tree. You've got ivermectin and penicillin that come from microbes. So it's not that much of a stretch when you think about the ocean and what the ocean can provide because it's such a rich source of biodiversity. So that biological diversity really underpins a really rich chemical diversity, and that rich chemical diversity is the potential for discovering novel chemicals or other biological products. And so the catchword for this is the blue economy. So what, in addition to compounds that sponges produce, are there other things that come from the ocean that can contribute to the economy? And so the blue, it's a real buzzword these days, the blue economy. And it's been estimated that the annual value of marine-derived pharmaceuticals is over $2 billion. So it's not insignificant. And there are a few drugs that are already clinically available that have come from marine organisms. So Prialt is like morphine, but it's about 100 to 1,000 times more potent than morphine. It's used really to treat pain in like patients who have cancer or chronically chronic pain management. And it's derived from a little cone snail, and the cone snail makes several different types of peptides or molecules. So each one has a different function. And one of the functions of one of these peptides is to deaden the pain of the fish that it's trying to eat. And so and there are many others. There are some that the cone snails produce that can cause paralysis. That's being developed into a drug that can be used for tremors. Okay, and then Yondelis is a drug that was derived. It's another cancer drug, and it was derived from a sea squirt that some of you probably have seen if you snorkel along and see it growing on mangrove roots. And in fact, the company that developed this initially collected a lot of the sea squirt from the Florida Keys. Sponges happen to be the most prolific source of marine-derived chemicals with pharmaceutical applications. And in fact, one of the first marine-derived drugs came from a sponge that was collected here in the Florida Keys in the 1950s. And the picture of the sponge on the far right, that glob that's got sediment all over it, that's a sponge that you'll see if you go snorkeling in grass flats. It's called a tethia or cryptotethia. And there are a couple of drugs that were developed synthesized based on chemicals in this sponge. One of them is an anti-herpes medication, so Zovirax. So if you get Zovirax to treat like fever blisters or herpes infections, that's based on a chemical that's produced by the sponge. And then there's another chemical that's used for non-Hodgkin's lymphoma. And then the sponge that's pictured on the lower left, I think it's from Japan, and somewhere in the Pacific. And Halaven is an anti-cancer drug. And there are many other chemicals that we call leads that are in the clinical development pipeline right now, mostly for cancer, but some that are like for infectious diseases, for malaria, for inflammatory diseases. And there's one actually that the group at Harbor Branch discovered many years ago that has anti-inflammatory activity, and it's also antiviral. So what might that be useful for? So the National Institutes of Health is taking another look at that particular compound. So why sponges? In most cases, we know more about what that chemical does in humans than what it's doing for the sponge. So what the role of that, why that sponge is producing that chemical, in most cases, we really don't know, because sponges don't get cancer, and we don't really know. It's not really well known, but the potential roles could include they're deterring predators, so preventing fish, other animals from eating them. And there are animals that eat sponges, sea stars, turtles, some fish, in fact, probably every kind of echinoderm eats a sponge. Preventing overgrowth, so preventing something from growing over top of it. How sponge is fixed to one place, how does it communicate with another sponge when it's time to reproduce? So there are some chemical signals that are going there. And then for sponges that live in very shallow water, there are some chemicals that we suspect are like sunscreens that protect them from UV radiation. So why would they have pharmaceutical potential? Now, I mentioned that sponges are ancient animals, but they're not primitive animals. So they have metabolic pathways that are very similar to the metabolic pathways that we have. And even though they don't have, eyes or a heart, they have metabolic pathways that they control processes in humans like that. So, for example, like cell division and cell death. So it's not far-fetched to think that a sponge might produce an anti-cancer drug, something that's going to prevent cells from dividing. If something's trying to grow over top of it, that sponge doesn't want that other organism to grow over top of it. It may produce a chemical that's going to kill those cells. Similarly, it might produce an anti-inflammatory compound, so when two sponges are touching one another, there's no inflammatory response. Calcium and sodium regulation, which would make it really interesting for discovering or developing drugs for heart disease, for example. More than 3/4 of sponge biodiversity is estimated to occur in deeper water, so below 200 meters. And remember what I mentioned to you, that biological diversity underpins chemical diversity. And that's what really gives us such a rich, kind of like pharmacopoeia, if you will, to discover new chemicals. And these are just some photos of deep water sponges that some that my colleagues have taken pictures of, and probably about half of them are ones that we've encountered. And so we've been really lucky at Harbor Branch Oceanographic, because for how many years, Don, did we have the Johnson Sea Lynx? Over 40 years. We had the Johnson Sea Lynx submersibles. I'm talking to my husband because he was a submersible pilot with the Johnson Sea Lynx. And it enabled us to go down 3,000 feet deep. And so we kind of had an edge over our competitors in terms of drug discovery, because we could go down deeper areas and really look for that rich biodiversity in deeper water. And we had very sophisticated collection tools. So instead of like trawling or dredging and picking up everything from the bottom, we could look at something and say, oh, that's really interesting. I was a great backseat driver. Go over there. I like that. Can you pick that up? Be real careful. Don't put it in the same bucket with the other one. And so we have over 30,000 samples that we collected over the years. So the whole process of doing that, and we had ships as well to be able, and that stopped in 2011, I think it was. So we had ships that took us, I mean, literally all over the world. Our drug discovery program focused primarily in the Atlantic, a little bit in the Pacific. We went to the Galapagos. We were on the west coast of South America, Central America. And so the way we would do this is you collect it could be a sponge or a soft coral or a sea squirt. And then we would prepare what's called an extract. And so, let me go, I'm going to go to the next slide. We prepare an extract. If you drink coffee or tea, you're making an extract every morning, because that you're preparing an extract of either the tea leaves or the coffee beans. Now, what you're looking for in that extract, for those of you who really need that caffeine kick in the morning, you're looking for that molecule. You're looking for the caffeine. That's what we're looking for. So that extract that we make of sponges, I usually call it a sponge daiquiri or a sponge milkshake, because we mix up the sponge, heat it up, and ethanol, alcohol. And it contains hundreds, or can be dozens to hundreds of chemicals. But what we're looking for is the caffeine. What we're looking for is that one chemical that's going to really do whatever it is that we're looking for. So if we're looking for an anti-cancer, we have specific biological tests or assays that are used to kind of determine how active that compound is. And so we have very skilled chemists who then can take those chemicals, so let's say the extract that has, say, dozens of chemicals, and then we test it to see if it's active in our assays or screens. And then they'll say, well, no, this one's not active, but this one is. And then biologists send it back to the chemist, and the chemist then say, okay, is it soluble in water or in ethanol and alcohol, butanol? Alcohol is a polar or non-polar. Take those two fractions, send the fractions back to the biologist. The biologist tests it and say, well, it's active in the aqueous or the water extract. Then goes back to the chemist, and little by little you get to a solution that has hopefully just one molecule, and that it still has the biological activity. Sometimes when you get to that one molecule and you test it in your assay, it loses its activity. So it's likely that it's some kind of synergistic activity. Now, pharmaceutical companies are not interested in that. They want a single molecule. They don't want to have to, I mean, even though you can take, have combination therapy where you may take two drugs to treat a disease, if you're trying to license that material for development, they don't want a mixture. They want a pure compound. And this is just an example of a fairly simple example of the type of test that we might do. So those little circles in there are like little filter paper discs, and you dip them into the extract, and then you put them onto this little plate. I'm poking to this like you can see what I'm doing. You put them on that little plate that's seeded with, let's just say, a pathogenic microbe. And then and it's clear at the time you start this test or assay. And then you incubate for maybe, let's just say, 24 hours. And after 24 hours, you look to see if there's a zone of no growth. We call it a zone of inhibition. And in the lower right, that would be the chemical on that filter paper disc is actually killing that bacteria or whatever it is that was seeded on that plate. And so that's what we're looking for, that type of activity. But it's not good enough to know that it kills a cancer cell, for example. You go to a pharmaceutical and come saying, I have this really great compound, it'll kill cancer cells. And they're going to say, okay, we have 50 of those. What makes yours better than the other 50 that are in our, in our portfolio? So you have to know what's called the mechanism of action. How does your drug that you want it to be a blockbuster, how does it do what you say it's going to do? How does it kill that cancer cell? So this is an example of a sponge that we collected, oh gosh, back in the late 80s. And the sponge is called Discodermia. And this one, it grows at about 400 feet deep. And this one I think was in the Bahamas. And several compounds were isolated from these sponges. And the ones that were really interested that we liked, we actually patented a license called discodermalide. The way discodermalide works, this is a picture. It's a picture that's taken with it through a microscope of lung cancer cells. And the green are what's called the microtubule network. So your cells have a little skeletal network of what are called microtubules that keep the structure of your cell intact. They're also, those microtubules are also really important when it's time for the cells to divide, because that's what forms the spindle that then the chromosomes move across so when the cells divide. So in untreated, and the red are nuclei. So these are stained with very specific stains that are targeted for either nuclei, so DNA, or these microtubules, tubulin. It's called tubulin. So this is what these cells look like if they're not treated. When they're treated with discodermalide, what happens is the microtubules stick together, and that means they can't form a spindle, and the cells can't divide. And to make matters worse, the nuclei start what we call blebbing. They start falling apart. So it's kind of a double whammy. So not only do we have like the microtubule network that's been disrupted, so the cells they can't undergo mitosis anyway, but then the nucleus is just all split apart. So this was a really important drug, and it was licensed to a pharmaceutical company called Novartis back in the late 90s. It got through a couple phases of clinical trials, and then they decided it was too toxic to proceed any further. It's being studied now at a much lower dose in combination therapy with, I think, the Taxol. So they backed off the dosage of both of those drugs to reduce that toxicity. And Taxol and discodermalide work in kind of the same way. The problem is that supply of these chemicals has been a bottleneck for clinical development. Why? They're generally present in very small amounts, and just trace amounts in the sponge. So, when you can't go and harvest them from the sea for a number of reasons. Number one, it's not ecologically sustainable, but also think about it. If we're going out, to distant locations on a ship with a submersible that's costing us in excess at that time of $25,000 or $30,000 or more a day, it's just not economically feasible as well. And these days you could never get a permit to do that wild harvest anyway. So it's really not sustainable, either ecologically or economically. Chemical synthesis of these molecules is actually the preferred route of a pharmaceutical company who really doesn't want to have to rely on kind of a biological production mechanism or going out and harvesting from the sea. They want to be able to synthesize the molecule. But these molecules are very, very complex. And so it takes, it can be multi-steps to chemically synthesize these molecules. If it's a really potent drug that's not, and for which there are no other drugs that are like it, pharmaceutical companies will spend the money to synthesize the drug. And so many years ago, because I'm a sponge fan, I worked on sponges all my life, and I really only have been working on biomedical applications of sponges for about the last, since 1984. When I was 5. Just kidding. Just kidding. Before that, I was more like ecological, but I've always been interested in sponge cells. And so when I joined Harbor Branch in 1984, I started thinking about, is there some way that we could, grow these cells in the lab and have them produce the chemical in the lab? I started thinking about that in 1990. So that started my quest to develop a marine sponge cell line. Why? Why cell lines? So a lot of times cell lines are used, like mammalian cell lines, like cancer cells, are used to help kind of discover drugs or to pinpoint a mechanism of action in drug. But I thought that this could be really useful as an in vitro or cell culture production mechanism. And it might also give us a new platform for different kinds of discovery of different molecules that we hadn't been thinking about. So not necessarily as anti-cancer drugs or antimicrobials, but something else. And that having a cell culture model might help us do that. And to help us better understand why these cells are producing the compounds. And then think about, because again, sponges are ancient animals, but with very sophisticated metabolic pathways. How did they evolve? How did they develop? what do we know? What can these cell lines tell us? So we started banking cells. So where we would take the cells and cryopreserve them, so at very cold temperatures, so that we could use them later on. And when we were working on developing these cell cultures, we'd go out in the field and, you could get fresh material. But then once I, went back to my lab in Fort Pierce, if I wanted that same sponge that was, say, down here in the Florida Keys, then I had to hop in the car and drive down. Not that that's not, it's pleasant to do that, but I can't do that. So I needed, plus you want to have a stock of cells that are all from kind of the same animal. So if you're doing experiments, you have, you don't have that individual variability. And more than 100 years ago, this man, H.B. Wilson, a scientist, and he was a cell biologist, but not a marine biologist. And back then, these biologists in the summertime would go up to Woods Hole and go to the Marine Biological Lab, and they putter around and do things. And H.B. Wilson one day left a sponge in a bucket of water and forgot that it was there. That is a no-no. And of course, it started disintegrating. But he realized that besides the real awful smell of the disintegrating sponge, that there were some cells that were actually aggregating, coming together and forming a whole new sponge. So it was really, and that's really what started kind of, thinking about cell cultures in general, not just sponge, in fact, not even sponge cell cultures. So that's what we started doing. And what you do is you just collect the sponge, you cut it into small fragments in a little petri dish in seawater. You use gauze to kind of squeeze it and you release the cells. And so they're all dissociated. We pour them through a little filter into a centrifuge tube and we spin them down and then we resuspend them in an antifreeze, a cryoprotectant. And then we dispense them into these little vials and then we freeze them down to -80 degrees. And you can go you can go much lower than that, but that works for us. -80 works for us. And so, after a while, the cells start. Okay, so this is a picture through a microscope and just looking at cells in a petri dish. And so you can kind of see some little cells in there. Now you can see they're starting to aggregate together, forming bigger clumps. Now they're starting to form a bigger structure. And now at a lower power, you can see they're all starting to send out little filaments, and all those are cells that are differentiating into different cell types. And here's a closer look at that. They're really spreading out. And now if you take a closer look at it, and I'm going to show you this. If you squint your eyes, you can see in the image on the bottom right, little circles, and those little circles are actually little choanocyte chambers. So this sponge now has started to differentiate and become functional. So the choanocyte chambers were these little sphere-like chambers with these collar cells that have the whips or flagella that are setting up a water current. And so it was like, okay, this is really cool.
What's the time frame between each?
That was about, I would say, less than a week.
Between each?
Yeah.
It's
Yes.
amazing.
Yeah. Don't get too excited because I got really excited about that. It's like, hey, good, we've done it because this was a while ago. Finally, after 30 years, we finally got this. The cells were dividing on a geologic time scale. I mean, slow. I am not a patient person. I want those cells to be dividing, and they would divide, and it's called doubling. Was they, one cell you get 2 weeks. Well, that's not going to help. If I want to make drugs, I'm not going to be able to sell that process to anybody. It's how do I need these cells to divide. So what happened? Sometimes it's just plain luck. These two sponges are the model sponges that we used for 29 of those 30 years. Great sponges. The orange one produces a chemical that's really easy to detect. It's called Stevenson. It's a common reef sponge that you see down here in the Keys. And the blue one is one that grows on mangrove roots. And it's similar to a sponge that occurs in the Mediterranean that produces an antiviral compound. We could not get those cells to grow, no matter what. We had created new nutrient media, like the little soup that we wanted to grow them in. Sorry, they wouldn't grow. So my student made a pretty good suggestion. One day she came in, she said, you know, we have this biobank. We've been collecting these sponges. We've been freezing them. We have lots of different species. Why don't we try the nutrient media on one of those? I'm like, knock yourself out. Try it. Lo and behold, the cells from this sponge divided like crazy fast. Like in the first, if you don't hurry up and take your cell counts right away, they've already divided. I would have never in a million years selected this sponge. It's hard. It has a thick cortex. I broke a knife on it 3 weeks ago, trying to collect a small piece of it and wedge it out. How about it? I broke my knife. That's how hard it is. But these cells are amazing. They just divide crazy, crazy fast. So now it's kind of cool because now we've got these cells that are dividing. And not only that, but because we started looking at our whole inventory, we've got lots of different sponges, just not the two that I worked on for 30 years. A lot of other ones that are really interesting. And so, but there are still some that don't divide. And so what do we do about that? And so we're working on developing methods where we can take cells from this sponge, fuse them with cells from the sponge that produces an interesting compound but won't divide, and make what's called a hybridoma, and then get that cell culture. So we've got a rapidly dividing cell that doesn't produce anything really interesting, fused with a cell that produces something interesting but doesn't divide. So that's a hot area of research in our lab right now. One of the other things that I'm not sure I have one of the other areas that we're working on right now as well is on habitat restoration. So we have a couple of projects that are funded by EPA, and we're looking at how we can create little sponges. So right now, little sponges that then we can use and create a sponge nursery, so that if there's an extreme weather event, so like when Wilma came through, when Irma came through, you remember, if you lived down here, there were like piles and piles of sponges that were washed up on shore. And so, we're looking to see how can we basically restore those habitats where they were before. So we're trying to create, notice I'm saying trying to create a sponge nursery. So we're getting the cells. So what we're trying to do, because right now the method that's used is, and it's a perfectly fine method, take the sponge, slice the sponge into, a healthy sponge, slice it into sections, fragments, attach those to blocks, put them out, in the bay, in Florida Bay. And they're doing well, but there was a call for proposals from EPA, and they were interested in some innovative methods. So we thought, well, maybe instead of taking a sponge, taking 100 sponges and making 1,000 out of it, like cutting, each one sponge into 10 pieces, maybe we can take a sponge and get billions of cells and then make little sponges out of those cells. And instead then of damaging a lot of otherwise healthy sponges, we can grow these small sponges. So we're working on that. We're 1 year into the project. Our biggest challenge right now, because the cells are dividing, we can get the cells to divide. We're working on loggerheads, and we're working on the base sponge, the Yersinia, it's kind of stinker sponge. And we're working on one of the commercial sponges, the sheep's wool sponge. We can get them to aggregate. They just won't make those dang choanocyte chambers and start filtering water. So that's our latest challenge, is trying to work on that. But we're getting there. We have some tricks that we're working on. I also have a student who's working on some other applications where she's using these cell culture models to be able to test the impacts of climate change. So she has, for her dissertation research, she selected 5 sponges that are common on the Florida Reef Tract. And she's testing them, and we've got cell cultures of them. It's the same student that was the one that said, why don't we try some of these other sponges in our inventory? And so we developed these models, and she's incubating these cell cultures at gradually increasing temperatures and gradually decreasing pH to see what the effect is on these sponges. And so just to kind of be able to predict, by the end of the century, based on the predictions of what the temperature is going to be, what's going to happen and which sponges are going to survive, which ones may not survive. So she's just focusing on 5 sponges right now, but we have a whole 2 freezers full with thousands of vials of cells that we want to look at overall, because these are the ones kind of like the iconic ones, like the barrel sponge and things that you think about when you go out on the floor or retouch. So she's working on that. So those are some of the areas that we're working on right now. So as our kind of the takeaway messages, sponges are dominant components of many marine ecosystems, from coastal environments to the deep sea, from polar regions to the tropics. They're important in carbon, nitrogen, and silica cycling. They are the most prolific source of marine-derived bioproducts, chemicals that have human health applications. And using the cell culture models that we've developed, we're testing hypotheses that are related to, as I mentioned, changing environmental conditions, being able to produce compounds in culture, and we're actually making very good progress with that. And then, as I mentioned, habitat restoration as well. So we have many sponsors who funded our research. I have many students who've worked with me over the years and colleagues. I have an appointment at a university in the Netherlands, and so we're working very closely with Renee Weifel's group and Dirk Martin's group in the Netherlands as well on this. And I recently have graduate students come to my lab from the Netherlands as well. We have a lot of sponsors who have supported our work. And our outreach program told me that I needed to show you this at the end of this. So in case you're coming up in the Fort Pierce area, we have a really nice ocean discovery visitor center that, and we offer tours of the campus as well. Harbor Branch was a private research institute that started in 1971. And in 2007, we became part of Florida Atlantic University. So we're affiliated with Florida Atlantic University right now. So Florida Atlantic is in Boca. Their main campus is in Boca. We're 90 miles north of Boca in Fort Pierce, but just a few miles off of I-95. So if you're heading north, please feel free to stop in and see our Ocean Discovery and Visitor Center. It's have really nice displays of all and kind of the work that we're doing there. I'd be happy to answer your questions now. Thanks so much for your attention. Any questions? Yes, Lisa.
I have a question about the chemicals. So you have to get the sample of the sponge, and then you have to get pieces of the sponge. So can it keep living while you chunk off little pieces?
Yes.
And test it,
So that's
chunk off little
well,
pieces
no.
and
Okay,
test
so
it?
that I don't know if you can I hear you when you answer the question over the? Okay,
Kind
let me
of reword
repeat.
it.
I'll repeat the question.
Yeah.
So the question was, when we're trying to get the chemicals from the sponge, can we grind up some and grind up the sponge and get some of the chemical out and then go back and grind it again and get. When we first collect the sponges from the ocean, the way we do that is we take the sponge, we take a small piece of it, like kind of a thumb-sized piece, grind it up in alcohol and freeze the rest of it. That was before Shirley got involved in the cell culture stuff, because now we take a piece of that and then we do the dissociation. So we just had a cruise to the Flower Gardens Bank in the northwestern Gulf of Mexico. And so out of I think they collected maybe, 120 samples, and we cryopreserved cells from, I think, about 50 or 60 of those. So we took a small chunk. So those are alive. So when we freeze those cells down to -80, we can thaw them, and then we can start doing that kind of work and saying, okay, so like, for example, one of the things I'm eager to try is, can we produce what's called unnatural natural products? What if we mix 2 sponges together? Will they produce something that they wouldn't normally do? People who do cultures don't like to do mixtures. But to me, it would seem like that would be kind of cool to do. We don't know yet what's going to happen with a hybridoma. It may be that hybridoma will refuse one cell with the other. It may not produce the chemical that we think it's going to produce, that we're hoping it's going to produce. It may produce something entirely different and maybe even better. So that's, that's a long answer. The answer to your question is no, they don't live unless I get my hands on them and cryopreserve them, and then they're alive. Yes.
What's the solution you use in the cryopreservation?
Okay, the question is, what solution do we use to cryopreserve the cells, the antifreeze? Because you have to prevent the cells from like we want them to freeze, obviously, but you don't want them to form ice crystals, because the ice crystals are what damage the cell components. And so we use a mixture of DMSO and yeah, DMSO and fetal bovine serum. That's what we use, DMSO and fetal bovine serum. We tested a bunch of things, and it's just a small percentage of that, 10%, like say in 100 milliliters, we have 10 milliliters of DMSO, 10 milliliters of fetal bovine serum. And then we freeze them very slowly. So we have these little containers that are called Mr. Frosties. We put them in the Mr. Frosty, and Mr. Frosty has like little receptacles for the vials, but underneath that is isopropanol. So that slows down the freezing process. And so we freeze the cells at 1 degree per minute. So to get from room temperature down to -80, it takes about like an hour and a half, something like that. And then we can keep them for years like that.
1 degree per minute, is that Fahrenheit or Celsius?
Celsius. Yeah, minus 80 Celsius.
Are
Yeah.
you taking your cryogenic ideas from Mary Haggerty in Hawaii?
Yeah,
Mary
so
Haggerty.
so the question was, are we getting the cryogenic ideas from Mary Haggerty in Hawaii? We've been doing the cryo, I don't know how long Mary's been doing this. She's actually been really successful at, I think she might be the only person, certainly the first person to culture coral cells.
This is true. This is why we got a chance to meet her and have a chance for her to share exactly how she was doing it as well. I was just wondering if you got that same process.
No, we've been using this process since 1992, I think.
Okay.
Started cryopreserving. We didn't start our cryobank until more recently. We were really just cryopreserving these darn Axonella cells just so we would have them to run our experiments. So we've been doing it for a while. I think Mary uses the same thing. In general, for animal cells, even if it's mammalian cells, you use DMSO.
And that's what she's using.
Yeah, and for microbes, they tend to use glycerol.
Mhm.
So we tried, I had a graduate student who experimented to see which was better, the glycerol or the DMSO, and the DMSO worked better.
Well, we do understand that the sponges are developing that type of habit with cryogenics. Is your idea same as the nematode? That's very well frozen in the Antarctic, but yet able to come back to life several years later
Yeah,
under the influence of nothing more than water.
right. So that makes you start thinking about space science,
Exactly.
right? Okay, so let me tell you a story. A few years ago, I got invited. Do I have time to tell a story?
Yes.
Okay. A few years ago, I got invited to participate in it was called a blue sky day. And it was a blue sky, like 2 or 3 days, talking about what transportation would be like 100 years from now. I have zero expertise in transportation. But the idea of this get together was to bring people from different backgrounds. And so there were astronauts there were guys that worked for the CIA, who were like, spies. And
Was Dennis Chamberlain part of that?
I don't think so. This was at the Institute for Human and Machine Cognition up in Pensacola. And I got into a conversation, and it was the last day. I got into a conversation with a guy who worked at Ames, Space Research Center in California. And he said, "Can sponges live in ice?" And I said, yeah, they can live in ice. He said, we should write a position paper on maybe sponges living, on these exoplanets. I said, you can get that stuff published. I didn't say stuff. I said, you can get that stuff published. And he said, yes, we can. We never followed through on that, but now I'm starting to think about that as well, because it's kind of interesting, especially since, we know that these, I don't want to say primitive, but early life forms can exist in very cold and inhospitable environments.
Well, they grow everywhere.
Yeah,
That's the
I know,
beauty.
very cold.
We do have a few questions that have come through from our
Okay.
Zoom. So
Okay.
Julia is going to let us know what's coming in.
Okay. Are they gonna, she gonna say? She's
Yeah.
gonna talk? Okay. Hi, Julia.
Hello, so we have two questions from Sally. Her first question is, how do you keep contaminants out of your sponge culture water?
Hi, Sally. We don't. So, oh, sorry. So the question was, how do you keep contaminants out of your cultures? Actually, I shouldn't say that. We use antibiotics. However, if you're trying to develop a drug, you don't want antibiotics in your culture. What we have found is that now that the cells are dividing, they don't get contaminated. So they must be in eating those bacteria. So we've been able, Megan's been able to wean our cultures off of antibiotics. So, but in general, because sponges, I mean, they're filtering bacteria, viruses, fungi all the time. And they're probably producing antimicrobials so that they don't get, they don't get microbial infections. But once you dissociate those cells, you have not only the cells, sponge cells, but you've got all kinds of other stuff in there as well. So part of it we can clean up a little bit when we centrifuge that suspension of cells. A lot of those bacteria stay in the supernatant, the part that doesn't come down in the pellet in the centrifuge tube, but we also add antibiotics. And the second question, is that also from Sally?
Also from Sally.
Okay.
This one's a little wordy, so bear with me. After initial trial and error to get consistent and successful juveniles grow out is one of the most consuming aspects of aquaculture. How long do you think it will take to get a sponge grown from a cell to planting size, and how much space would it require in the lab, or could it be done in the sea?
Okay, so I'm gonna distill that. It takes a long time to get something to grow from like a single cell stage or a few cells to something that you can outplant, for example. I know I'm making it a little bit simpler than what Sally had asked. So how long would it take? Remember, I'm an impatient person. I can't wait for them to divide on a geologic timescale. So what we're hoping to do is to get them to kind of stimulate them so that they'll aggregate and form these structures. And we're using matrices, like little filter paper, kind of like filter paper discs to get them to grow, or we embed them in like in a gelatin, for example. I'm going to start trying to do something else where I'm just looking at maybe taking a plug. So I've already got the sponges that are already differentiated, and we already know that if you cut a sponge, it grows, it can heal pretty quickly. So my next, my next step is to actually look at, see whether we can take some plugs and grow those. But to answer your question, Sally, I think that it's going to probably to the point where you can get them into like a hatch, like an aquaculture situation. I think they just have to be, like maybe like the tip of, like not even the tip of your pinky finger, as long as we can get them so that they're not, moving around. How long would it, how much space would it take? It could take a fair amount of, think about how much space like the coral nurseries take. And so our goal would be, once they're big enough to outplant and to move them out to the environment right away. But you don't want something that's bite-sized, that a fish can come and just snip off right away, or a turtle or a starfish. So they're going to have to grow, I would imagine, probably like golf ball size, I think, before we can outplant them. So it'll, I personally, I think it's going to take maybe a year or so to get them to that size. But the answer to that question is, I don't know. That's what we're trying to find out. And we have a question in the back. Yes.
You mentioned you looked at the temperature tolerance and the acidity tolerance of 5 species on ocean side. Have you looked at any of the species on bay side, like loggerheads and bases?
Right.
That's a lot shallower, and they deal with the cyanobacteria blooms too.
Right. So the question is, because I mentioned that my graduate students, did you wonder the question? Was that the question?
That was a question.
Okay. So the question was, my graduate students were looking at reef sponges, and the question was, are we looking at sponges in the bay? And the answer to that question is yes. In fact, those are the ones we're really focusing on for growing for our sponge nursery. We're really focusing on the bay sponges for our sponge nursery, that was what the call for proposals was from EPA. So that's, and it's kind of hard to plant stuff out on the reef. So while it'd be nice to be able to say, yes, we're growing the barrel sponge and stuff, we're really focusing on it, on being able to grow the bay sponge. You know how hot that water gets. It gets really hot. And so what we've found is that they're fairly temperature tolerant and pretty irritating as well. So we, like, for example, when we embed the cells or immobilize the cells in gelatin, we have an ultra low temperature gelatin, but we still have to, it's for it to be liquid, it's still got to be 37 degrees C. So how do you, are they going to stay alive? And the cells actually do stay alive, even the reef sponges. Yes.
A question is how can citizen science get involved in sponge restoration?
So, and it's something that I've just been talking about. We were just talking about it recently, because I think that if this process that I'm considering doing now is where we take little plugs out and then start them off kind of in a raceway situation, but kind of embed them in some kind of substrate where we can get them to start growing. That's where we're going to need citizen scientists, divers and snorkelers who can help us, actually do that sampling. And then certainly when it's time to do the outplanting, we're going to need help, because we're a small group, and so we really depend on volunteers. Shirley, I know you're really interested in citizen scientists as well. So Shirley Kruger's got one of the EPA grants that we're working on, and she's really interested. And she spoke at one of the lectures earlier this season. And so, I know that she's hoping also to be able to use citizen scientists for that research as well. Yeah, we're going to need help when it comes time. If this works and we're producing a lot of little sponge babies, we're going to need help getting those babies out of the nursery. Yes.
What if,
Oh, sorry.
what if I had a bay nearby in the Keys that was available to be able to have already a little small coral reef that has some sponges that could be grown on even more? Would that be helpful to you for more citizen science?
Oh,
I'm a
sure.
science diver.
Yeah, sure. Pilings, your docks, things like that. And you're kind of doing that in the lower keys as well, right? So yes, that would be very helpful. As Pomponi at FAU.edu.
I was going to ask you a question because we're sharing the pad. Are you affiliated with iCare? Do they help you with donations?
With
I care.
No, I'm not
You haven't
affiliated with
done with
iCare. I'm not
Oh, I
affiliated with
thought it was you
iCare.
and your students. It must have
Okay,
been
as
another
Pomponi
doctor.
at FAU.edu. Get in touch with me.
Yes.
Yeah, because see, iCare has already received quite a bit of funding, nothing more than coral restoration. So
Okay.
But
we
they're
are-
in as well
They work
over
with
the
another
summer.
team
Yeah.
from FAU.
Is that, are they at University
They're
of
in
Miami?
Alamorada.
Oh,
Right.
Colorado. Oh, nice. Okay.
St. Mary's.
Yeah.
Right,
Okay.
but
Okay.
they developed it so that there was funding that came in where all the locals have an opportunity of going out and regrafting coral into the reefs that are already here.
Outplanting.
And so
The
the
coral,
outplanting itself,
yeah.
Bioengineering the reefs
which is
back to
pretty
health.
much what
Right.
you're looking to
Right.
have done, I can tell.
Yes.
And we're working with sponges as well with another team from FAU.
Do you know what sponges you're working on? Is it
I don't know which
loggerhead or?
species they were working with.
The ropey ones, what color? Blue?
Red.
One was red and one was, they're also working on green.
Yeah, green.
Red, okay, I know what those, okay, the red ropey one is that's a fairly common
common
reef
here.
sponge.
Yeah.
And the green one is the green one, when you squeeze it, black stuff comes out?
Yeah.
Yeah, those are really good ones. They're, those ropey ones grow pretty well.
Well, if you're looking to see how these guys are doing it, I want to talk to these guys.
Okay, I will. I'll definitely
I'll
do that.
connect.
They're probably cutting fragments from them.
Yeah.
And then doing that. So what we're trying to do with the cell culture is to avoid taking an otherwise healthy sponge and then fragmenting it. So what we're hoping to do is just take small pieces of it, the sponge heals, and then we've just got the cells from that sponge. So that's kind of the approach that we've been taking. And and there are many different approaches to it. And I know that for many years, Mark on the group from FWC and DEP planted 15,000 sponges in Florida Bay. Unfortunately, a lot of them got damaged
Turn
from
on.
from Irma. And then every time there's a harmful algal bloom, phytoplankton bloom, some of them don't do very well. Is there another question?
No, we were just checking online. We're good
Okay.
online. Any more in the audience here?
Just one person who left a very nice comment. This has been a very fascinating talk, and thank you.
Oh, thank you. Thanks. I love talking about sponges.
Do you work with Mark Butler or Don the manager at all?
We don't, I know who they are.
Okay.
So, yeah, in fact, we're just talking about this because we applied for a grant, an EPA grant. And I asked him for like a letter of support. He said, "Oh, we're going to be competing against each other." And I said, why don't we just write letters of support for each other? So, yeah, they've done some really nice work, really nice work. They're the ones that did all of those, I think, right? Aren't they the ones that planted all the 15,000, along with John Hunt's group and,
Yeah,
that
Mark
group and
Butler was
Mark
on that
Butler.
grant.
Yeah, yeah.
Well,
really nice.
The gentleman over there at iCare, his name's Mike Goldberg.
I'd love to get in touch with them.
Okay.
So I'll leave my. I will
Okay,
send
great.
an
Thank
email.
you. I appreciate that.
So
Yeah,
if anybody here wants to get in touch with you, give everybody and on Zoom your contact information again.
Okay, S Pomponi, so SPOMPONI at FAU for Florida Atlantic University .edu. I should have put that up here.
Actually, it was up there in the beginning. I believe.
Yeah.
Yeah.
So thank you very much
You're
for
welcome.
coming.
It's my
We
pleasure.
appreciate you
Thanks.
being a member at the History of Diving. It was really great working with our team,
Yes.
putting together the Sponge Hunters exhibit.
Yeah,
So we
that
thank
was
you and
fun,
thank you
yeah.
for your use of sponges and artifacts to add to it as
You're
well.
welcome. It's my pleasure. Thanks.
Uh-huh. So we have a nice little gift bag for you. She got a
She
Sponge
already gave
Hunters.
you a
We gave you a
t-shirt.
t-shirt earlier
Oh my
today, so
gosh.
we've got a little thank you
Oh, thank you
package.
so much.
And again, thank you for being a member of the History of Diving Museum. For those of you in the audience, you can reserve a seat. If you're online and you're watching this through our YouTube channel, you can always add questions to the comment section, and then we can
Oh, yeah, if you just
reach
send it to me,
out
yeah.
and get back to you.
Yeah,
So
Is there
sounds
any
good.
prevention in there?
is there any what?
Prevention?
No, there's
I think that's done.
Thanks for coming tonight. We look forward to tuning in next month on the third Wednesday.
Great, thank you.
Thank you.